me again, Erica Moore, and welcome to my YouTube channel. So I am back again for another discussion, and for today's video, I am going to tackle about the protein synthesis, specifically the transcription. DNA or deoxyribonucleic acid is the molecule that codes genetic information. However, DNA can't directly order a cell to make proteins. It has to be transcribed into RNA or ribonucleic acid. RNA, in turn, is translated by cellular machinery to make amino acids, which it joins together to form polypeptides and proteins. Let's take an overview of transcription. Transcription is the first stage of the expression of genes into proteins. In transcription, an mRNA, messenger RNA, intermediate is transcribed from one of the strands of the DNA molecule. The RNA is called messenger RNA because it carries the message or genetic information from the DNA to the ribosomes, where the information is used to make proteins. RNA and DNA use complementary coding where base pairs match up, similar to how the strands of DNA bind to form a double helix. One difference between DNA and RNA is that RNA uses uracil in place of the thymine used in DNA. RNA polymerase mediates the manufacture of an RNA strand that complements the DNA strand. RNA is synthesized in the 5 to 3 direction. There are some proofreading mechanisms for transcription, but not as many as for DNA replication. Sometimes coding errors occur. There are significant differences in the process of transcription in prokaryotes and eukaryotes. In prokaryotes, bacteria, transcription occurs in the cytoplasm. Translation of the mRNA into proteins also occurs in the cytoplasm. In eukaryotes, transcription occurs in the cell's nucleus. mRNA then moves to the cytoplasm for translation. DNA in prokaryotes is much more accessible to RNA polymerase than DNA in eukaryotes. Eukaryotic DNA is wrapped around proteins called histones to form structures called nucleosomes. Eukaryotic DNA is packed to form chromatin, while RNA polymerase interacts directly with prokaryotic DNA. Other proteins mediate the interaction between RNA polymerase and DNA in eukaryotes. mRNA produced as a result of transcription is not modified in prokaryotic cells. Eukaryotic cells modify mRNA by RNA, splicing 5' capping and addition of a poly-A tail. Transcription is a process in which information is rewritten. Transcription is something we do in our everyday lives, and it's also something our cells must do in a more specialized and narrowly defined way. In biology, transcription is the process of copying out the DNA sequence of a gene in the similar alphabet of RNA. Steps of transcription. The process of transcription takes place in the cytoplasm in prokaryotes and in nucleus in eukaryotes. It uses DNA as a template to make an RNA, messenger RNA molecule. During transcription, a strand of messenger RNA is made that is complementary to a strand of DNA. Transcription uses the sequence of bases in a strand of DNA to make a complementary strand of messenger RNA. Triplets are groups of 3 successive nucleotide bases in DNA. Codons are complementary groups of bases in messenger RNA. Transcription takes place in 3 steps: initiation, elongation, and termination. Step 1. Initiation. Initiation is the beginning of transcription. It occurs when the enzyme RNA polymerase binds to a region of a gene called the promoter. This signals the DNA to unwind, so the enzyme can read the bases in one of the DNA strands. The enzyme is now ready to make a strand of messenger RNA with a complementary sequence of bases. Step 2. Elongation. Elongation is the addition of nucleotides to the messenger RNA strand. RNA polymerase reads the unwound DNA strand and builds the messenger RNA molecule using complementary base. During the process, an adenine in the DNA binds to an uracil in the RNA. Step 3. Termination. Termination is the ending of transcription and occurs when RNA polymerase crosses a stop sequence in the gene. The messenger RNA strand is complete and it detaches from DNA, pre-RNA and mRNA. After transcription, eukaryotic pre-mRNAs must undergo several processing steps before they can be translated. Eukaryotic and prokaryotic tRNAs and RNAs also undergo processing before they can function as components in the protein synthesis machinery. mRNA processing. The eukaryotic pre-mRNA undergoes extensive processing before it is ready to be translated. The additional steps involved in eukaryotic mRNA maturation create a molecule with a much longer half-life than a prokaryotic mRNA. Eukaryotic mRNAs last for several hours, whereas the typical E. coli mRNA lasts no more than 5 seconds. The 3 most important steps of pre-mRNA processing are the addition of stabilizing and signaling factors at the 5 and 3 ends of the molecule, and the removal of intervening sequences that do not specify the appropriate amino acids. 5. Capping. A cap is added to the 5 end of the growing transcript by a phosphate linkage. This addition protects the mRNA from degradation. In addition, factors involved in protein synthesis recognize the cap to help initiate translation by ribosomes. 3. Poly-A tail. Once elongation is complete, an enzyme called poly-A polymerase adds a string of approximately 200 A residues called the poly-A tail to the pre-mRNA. This modification further protects the pre-mRNA from degradation and signals the export of the cellular factors that the transcript needs to the cytoplasm. Pre-mRNA splicing. Eukaryotic genes are composed of exons, which correspond to protein coding sequences, and intervening sequences called introns, which are removed from the pre-mRNA during processing. Intron sequences in mRNA do not encode functional proteins. All of a pre-mRNA's introns must be completely and precisely removed before protein synthesis. If the process errs by even a single nucleotide, the reading frame of the rejoined exons would shift, and the resulting protein would be dysfunctional. The process of removing introns and reconnecting exons is called splicing. That's all for this video. So I hope you learned a lot about our topic today. And thank you for watching, and see you on the part 2 of this topic. So thank you for watching. Bye.